Hi, it's great to have you here. My name is Dimitria Brempou, and I will be presenting my Parenclitic network approach for extracting information from DNA methylation data and how I applied it to classify aggressive neuroendocrine cancer. My research focuses on pheochromocytomas and paragangliomas, a rare type of neuroendocrine cancer. Despite the high degree of heritability, PPGLs present great phenotypic variability. So far, there are no markers or tools able to predict the phenotype. For the work I'm presenting today, I used two different independent datasets: the Cancer Genome Atlas dataset available in the public domain and an unpublished dataset created in our lab. DNA methylation is an epigenetic mechanism regulating gene expression, and it is often perturbated in cancer. In a nutshell, when the promoter region is methylated, the transcriptor factor cannot bind, causing the silencing of the gene. There is a subset of PPGLs which present global hypermethylation. The same subset also presents a higher risk of developing aggressive disease. This is what made us wonder whether the DNA methylation patterns of the tumors can reveal its aggressive potential. To answer this question, I utilized a method proposed by Zanin et al. In 2014 called Parenclitic networks. This method allows us to study the deviation of the DNA methylation of the aggressive samples from that of the non-aggressive samples. This is an agnostic approach, which takes into account gene interactions, and by studying the topological features of the graph allows us to overcome the small n big K problem. Moreover, due to the way the graphs are constructed, they form these hubs, which offer further insights into the underlying mechanisms and the drivers of the differences we observe. I used parenclitic networks of the DNA methylation status of each sample, extracted the topological features, and used them to classify the phenotype. More specifically, I used 141 randomly selected non-aggressive samples from the DCGA cohort to build a model for the DNA methylation of non-aggressive PPGL. I modeled the behavior of the DNA methylation in each pair of loci using a Gaussian mixture model. For the training, I used the remaining non-aggressive and all the aggressive DCGA samples. Each locus is represented by a node, and the edge connecting each pair of loci is weighted by the distance from the model. After building the graphs, I extracted the topological features and I used them to train a classifier. I used different topological features and classification methods, and I found the best performing classifier to be bagging of logistic regression classifiers. For the testing, I used our local independent dataset and followed the same approach. Visualizing the graphs already highlights great differences between aggressive and non-aggressive samples, with the aggressive ones, as expected, being represented with more well-connected graphs. This difference is also reflected in the topological features. These box plots show the node degree in different loci for aggressive samples in orange and for non-aggressive samples in blue. Using the node degree, our classifier achieved balanced accuracy of 78%, and considering the lack of predicting tools for this type of cancer, these results provide a first point of reference based on DNA methylation data alone. This approach highlighted the DNA methylation perturbation in aggressive PPGLs compared to non-aggressive ones, and has offered new insights into the epigenetic landscape of the disease. If you want to hear more, reach out to me through email or the Juno app. Or if you are attending the conference in person, you can find me in the poster session B on Tuesday the 25th, and my poster number is 319. With this, I'd like to thank you for your attention and my lab as well as my funders for all their trust and support.